Yeah, good afternoon, everyone. A warm welcome to the speaker and all the participants and all the audience here. My name is Ravi Kiran, and I am a member of the Science and Technology Council. And this year, Anvesha is proud to be sponsored by Sabari Scientific Supplies, Science Apparatus and Chemicals. As you all know, we have organized this Nobel Lecture or Nobel Lecture series in collab. Today's talk is in collaboration with the Chemical Hub of ISER, and we are happy to invite the speaker. And I request Jahnavi of the Chemistry Club to introduce today's speaker and continue with the talk. Thank you.
Thank you, Ravikiran. Hello, everyone. This is Janvi on behalf of Chemistry Hub of ICTVM. First of all, I would like to welcome all of you to today's session. Here we are going to have the first talk on the Nobel Prize talk series organized by Anvesha, the Science Club of ISER Trivandrum, in association with Chemistry Hub of ISER. This year, the Nobel Prize in Chemistry has been awarded to Dr. Emmanuel Charpentier and Dr. Jennifer A. Doudna for development of a method for genome editing, or the CRISPR-Cas9 system, that is, the clustered regularly in the space short palindromic repeats on the CRISPR-Cas9 technology. I would like to introduce our speaker, Dr. N. Sadananda Singh from School of Biology, ISET Trivandrum. He holds a PhD in microbiology and cell biology from one of the premium science institutes in India, the IISc. His extensive research in investigating the genetic factors and mechanism behind biological systems and the development of CRISPR-Cas technology and its application in genome editing to recognize genetic factors behind drug responses is truly inspiring. Today, we will revisit the Nobel Prize winning work in CRISPR-Cas9 genetic scissors. Our speaker, Dr. N. Sadananda Singh, will be giving you an expert exposition on the research and the development of a method for genome editing. Once again, I welcome you all, and I welcome Dr. N. Sadananda Singh to start the presentation. Thank you.
The opportunity to talk about this technology that I'm very much excited about in my research work as well, and more importantly, to share you the excitement of awarding two of the pioneers in this field with a Nobel Prize. So I'll start. And how it go? Okay, see the slides, correct?
Yes, it is visible.
Yeah, okay, great. So we are going to talk a little bit very basic about CRISPR-Cas technology today, which was discovered by Emmanuel Charpentier and Jennifer Doudna. And the technology has been impactful in wide-ranging fields of biology, and it has been awarded or recognized with the Nobel Prize for 2020. So from the Nobel Prize Nobel website, it is cited the reason for awarding the Nobel Prize is for the development of method for genome editing. So before we go into genome editing, I just want to brush very basic of biology, how gene works. So if we look into the genome as such, it is stored in a string of the nucleotides, that is the DNA. Okay, and DNA, this string is consists of 4 letters, that is the thymine, adenine, cytosine, and guanine. So these 4 letters can be arranged in a series in a very different pattern and stores the information about how the organism is going to develop, how the organism is going to behave in certain environment, or otherwise how a disease is going to be caused. So if we look further into DNA, how this stored information is passed on to give rise to the signals that we see, the phenotype we see is that the string of DNA is copied into a molecule called ribonucleotide into RNA. And this RNA, if you look at it, it is just very similar to DNA, except that the sugar moiety is ribose, not the deoxyribose in the case of DNA. And it is copied or transcribed into RNA. And this RNA, if we look into further how it gives rise to a signal for the phenotypes or the whatever you see in an organism is through these molecules. So DNA is being transcribed into RNA. Once RNA is formed, for these RNAs can be divided into two. One is the protein coding genes or protein coding RNA and non-coding RNA. In the case of protein coding RNA, what we do is that there is a machinery called ribosomes. Ribosomes help in reading the RNA in three bases. Okay, here for example, GUA is read as amino acid as valine, and UGC as cysteine, glycine, serine, and leucine. So like this, triplets of the bases are read from the RNA to synthesize the protein. Now coming to, if there is a change in the sequence of the bases in the DNA when it is transcribed, that will be transferred into RNA. When RNA is being read, for example, in this case, from AAG, which code for lysine, if it is changed or mutated to form CAG, then it will give rise to glutamine instead of lysine. Okay, so why I'm telling you here is that the changes in the DNA sequences, the order in which the letters are arranged, it's being changed or mutated, then it can give rise to changed protein, which can have different functions or faulty functions. And so to give an example here is that there is a protein called beta globin gene in the blood cells. When it is expressed in a normal scenario, one of the codon is GAG, which codes for glutamic acid. In certain situations, this GAG is changed to GTG, causing a mutant protein having valine as amino acid instead of glutamic acid. This has a profound impact in such that the red blood cells in the system in our body, red blood cells, which has a donut shape structure, is changed to a sickle shape, and these changed sickle shape cells cannot transport oxygen from the lungs to the different parts of the body. So it has a very, very profound impact on these mutations can have on the system. So then the next question arises. If this kind of mutations are found in an individual, then it will be very, very beneficial if we can change these defects in DNA. For example, when the both the strands of DNA is broken to give rise to double strand break, what you can get is that the system can repair this double strand break into by two modes mainly. So one is the non-homologous end joining method, wherein the broken ends are just glued together so that it just continues to have these intact DNA. However, this pathway is error prone, so it inserts or delete bases from the DNA. It's just glued together, whether there is error or not, it's just glued together so that DNA structure is intact. However, there is another very high fidelity repair system called the homology directed repair HDR pathway. Herein, it uses another copy which can be utilized to repair this broken DNA. So here, the homology directed repair being error-free, it is very, very useful if you provide from your if you provide a template of correct gene sequence. For example, the defect I just cited for the sickle cell anemia, if you provide a template of DNA which has the correct sequence of that beta globin gene, what it can happen is that it can correct the sickle cell anemia mutation, and you can have a normal mutation, normal gene which can function well, and it can have a positive impact on the individuals. So people have been exploiting, but the difficulty is that how to bring about a double strand break where it is desired. That is the main question. Earlier, people used to introduce random breaks, and it was and try to use this homology directed repair, and you can get the normal genes. But it is very, very less efficient, and it used to take years. For example, just to correct a mutation, it used to take maybe 5 years or PhD just to correct that mutation. So with this importance to have a system to introduce double strand break at a desired location wherever we wish. If we can introduce double strand break, you can provide the templates to have the desired changes that we want to have. That is called the genome editing technology. So before the CRISPR-Cas system that we are going to talk now, there were other genome editing technologies started from the very bulky and very difficult to engineer, that is the meganucleases. Then came the slightly easier zinc finger nucleases, then very modular, very advanced protein engineering talents had come before the CRISPR-Cas system. If you observe here, all these are based on protein activity alone. So these were very difficult to engineer in a large scale setting because it takes a lot of time and it takes a lot of purification, all this, and it used to be very, very costly. But CRISPR-Cas technology transforms the technology that you will see how it has transformed this technology, genome editing technology now. Okay, so going ahead, I will just give you a brief history how it has come to this development of CRISPR-Cas system. Is that CRISPR-Cas system started with a very simple observation during the study of a gene called alkaline phosphatase enzyme in E. coli, that is the lab bacteria that we use very often. And when they were looking at the alkaline phosphatase enzyme, isoenzyme, in that locus, what they have seen is that just downstream of the gene for alkaline phosphatase, they saw a repeat of sequences. Here it's starting from CCC. They could see 5 of them present next to the alkaline phosphatase gene, isoenzyme. So they thought this is kind of a repetitive element, and they didn't know what is the function of it. So that was the first observation of having a repeated element in the genome of E. coli. Then came people, a lot of bioinformaticians came and then looked into this system and found to be present in many, many other bacteria as well as archaeal system. The basic structure, how these repeated elements is arranged is that R is this repeated element that has been observed in the alkaline phosphatase locus, R, which is constant. And if you see, these repeated elements is spaced out by a sequence which is variable from one repeat to the other. So here in this example, S1, S2, S3, S4, these are called spacer elements. And these spacers are different or unique in each case. Then these repeat elements, if you look at it, because of the nature of its arrangement, it has been called CRISPR in short form, and in the expanded form is called the clustered, regularly interspaced, short palindromic repeats. Okay, so you have a clustered repeats, and you have these repeats are spaced out by unique sequences called spacers S1, S2, S3, and S4 in this case. The term CRISPR was first used by Ruth Jensen in 2002, and that time people didn't know what the CRISPR does. They just observe it is present in many organisms. This kind of repeats are present in many organisms, but they didn't know what is the function of it. Then there came elegant study from including people like Mojica. When Mojica was the postdoc in Elena Soria's lab, what he had observed is that these spacer sequences that we talk about, S1, S2, S3, and S4 in this case, these sequences are derived from already present prokaryotic or bacterial sequences, or bacterial virus sequences that call bacteriophages sequences. So when he looked at it, it is already derived mainly from bacteriophages. Bacteriophages are the viruses of bacteria. So when he looked at it, what he has found is that you have the CRISPR array here, the repeat elements with black diamonds here, and spacers with different square boxes here. It was arranged like this, and just near to it, there are genes called CRISPR-associated genes are present in all the systems, either ahead of this CRISPR array or downstream of it is always present. And another observation which was very interesting made by Mojica was that if the spacer sequence is matching with a genome of a bacteriophage or virus of the phage, this bacteria is protected against this particular bacteriophage. For example, here green, so this bacteria is protected from the phage which have this sequence. So they concluded that if this CRISPR array, they concluded that this CRISPR array is somehow related to the immune system of bacteria, and because it is keep on changing depending on the phages which are present in the system or being infected, it is termed to be an it is considered to be an adaptive immunity. So CRISPR array is associated with adaptive immunity of bacteria. That was the observation. Till then, how this CRISPR array and Cas9 function was not well known. At this time, other people in the bioinformatics field also looked at it, and they tried to classify the CRISPR-Cas system. The Cas proteins, if they look at it, they classify into two classes, that is class 1 and class 2. How it is classified is that the class 2 system is much more simplified, mainly the protein which has the nuclease domain. Okay, nuclease are the enzymes which cleaves DNA or RNA. The enzyme which contains this nuclease domain is single unit in the case of class 2. So the whatever immunity that we are talking about, it brings about by a single enzyme in the case of class 2, and class 1 has multiple proteins to bring about this immunity. Okay, so that was the observation, and many people were looking at the class 1 and class 3, but here comes the Emmanuel Charpentier. She was interested in bacterial infection and how immunity, or how does it cause disease, or how bacteria gains immunity. That was her interest, and what she had observed is that there is a small molecule. She was interested in small RNAs which are involved in bacterial infection system or the immunity as such by in the bacteria. So what she had observed was that there was a small RNA consistently present in Streptococcus pyogenes. Okay, Streptococcus pyogenes, and these RNA, she called it as tracer RNA. Okay, this tracer RNA is sitting just ahead of the Cas proteins. Okay, so these Cas proteins, along with tracer, works together to give rise to the maturation of these CRISPR RNA. Okay, what she has found is that she had discovered this small molecule, small RNA called tracer RNA, which is involved in getting the CRISPR locus transcribed into a single RNA, and it is being processed to give a mature CRISPR RNA. So she discovered this tracer RNA. Still then, there was not much known about mechanism how this act as a adaptive immunity for the bacterial system. Then came, according to Jennifer and Charpentier, what they have tell that they met in a microbiology meeting, okay, in Europe, I think. Then they talk about the system. There is an immunity, and mainly Charpentier was talking about this small molecule tracer RNA to Jennifer Doudna, and it is somehow linked to bacterial adaptive immunity that was told to Jennifer Doudna. That's where they set up a collaboration between them, and they try to work out how it is working at a molecular level. That's where they got this publication in Science in 2012, and this is what led to the Nobel Prize for these two scientists actually. So what they have looked at is, what they have found out is that tracer RNA has a base pairing or complementary region in the CRISPR repeats. So these CRISPR repeats, along with the tracer RNA, forms a complex with one of the protein called Cas9 in Streptococcus pyogenes. And this Cas9 protein has two nuclease domain. Nuclease domain 1, that is the RAFC domain, and another domain called HNH motif. Both these nuclease motifs or domains can introduce and cut in the DNA in such a way that it depends on the CRISPR array spacer sequence that I talked about. If the spacer sequence match with the target DNA, then it can introduce these DNA cut on both the strands of DNA, using the RAFC motif and HNS motif in the DNA. So now, what they have observed also is that initially they used two separate RNAs, the CRISPR RNA that they use in the they found in the CRISPR array, along with tracer RNA. Two separate RNA were used to introduce these double strand breaks. And in the same paper, what they have also found is that if you join these and synthesize a single RNA, it still works as nuclease and giving rise to double strand break in the target DNA wherever the spacer sequence has complementarity. So that was the first discovery they have found. And then Jennifer was very much involved with this study, and she worked out furthermore what specificities are there in these RNA. What they have found out is that wherever there is a specific nucleus cutting is there is a certain motif in the genomic DNA or target DNA. This is called proto-spacer adjacent motif, PAM. Okay, PAM sequence. So what she has found out is that wherever there is a target, wherever there is PAM sequences along with a spacer matching region or proto-spacer in the case of genomic DNA, the enzyme and along with the complex, that RNA complex that is called now the guide RNA. Okay, this guide RNA along with the Cas9 binds more tightly to the target sequence if the target sequence has PAM sequences. So PAM sequences influence how long the Cas protein along with the guide RNA sits on the target DNA. It has a lot of implications. How it is? Because if there is a PAM sequence, then you can see DNA cleavage. But if there is no PAM sequence in the target DNA, there is no nuclease activity of that Cas9 along with the guide RNA sequence. So somehow the PAM sequences is involved in recognizing or activating the nuclease activity of Cas9 along with the guide RNA complex. So further, Jennifer looked into the crystal structure along with and tried to find out how these PAM sequence binding and title binding sequences help in recognizing the target sequences and introducing the nuclease activity later on. So what she has observed from the crystal structure is that once the Cas9 along with the guide RNA binds to the target sequences. If there is PAM sequence, the complex opens up or melts the double-stranded DNA and allows annealing of the spacer sequence to the target sequence. Once this annealing with spacer and the target sequence happens, there is a lot of dominal changes, or there is a conformational changes on the protein in such a way that the HNS motif as well as the RAFC motif comes near the cleavage site and introduces the double strand break. So when she looked at this crystal structure, what finally comes out is that the Cas9 protein along with the guide RNA sequence comes and probes the DNA in the genome. Wherever there is a PAM sequence, it tries to melt and allows annealing of the spatial sequence. When there is a spatial sequence matching, it brings about a conformational change on the Cas9 enzyme and introduces the double strand break at the target site. So now coming back to how this CRISPR-Cas system is involved with immunity of against the bacteriophage, because it has its own relevance. How it is that when there is an infection by a bacteriophage and the bacteriophage injects its genomic DNA inside the bacteria. Once this genomic DNA of bacteriophage enters the cell of inside the cell, what happens is that some of the Cas proteins allows cleavage and take it to the locus, the CRISPR locus, and insert it here and kept it as a spatial sequence. So when there is a first infection, when there is infection, part of the genome of the bacteriophage is inserted as spacer in the CRISPR locus, that CRISPR array we talked earlier, and it is stored there. And it is transcribed and processed along with tracer RNA and the RNase tree of the host system. And you get these small RNAs with repeat elements, which has a structure, repeat elements, and the spacer sequences. So now this spacer sequences matches with the bacteriophage sequence. Now it is to be noted, when there is a bacteriophage infection for a second time, then when the bacteriophage injects its DNA, what happens is that the Cas9 along with this CRISPR guide RNA, which is already present, and it matches with the genome of this bacteriophage, it goes and finds the target site and cleaves, degrades the DNA of these bacteriophage. So by degrading the genomic DNA of the bacteriophage, it protects itself from phage infection. That's how the CRISPR-Cas system was involved in protection against bacteriophages. Going ahead, to summarize what we have talked about here is that there is a RNA molecule. Earlier when it was before the engineering, it used to be 2 RNA molecules. Now it is a single RNA molecule called a single guide RNA, sgRNA here, and with the protein Cas9 forms a complex and goes and finds in the genomic DNA the sequence matching to the spatial sequence. Once there is a match and you have a PAM sequence, it introduces double-strand breaks. Once the double-strand breaks are introduced, what happens is that if you provide a correct sequence, another copy, the defective gene can be repaired, or the cell can go just to glue this double strand back together with errors in that. These errors give rise to the non-functional protein or non-expression of the gene. So this helps in getting rid of genes, and you can study the function of the genes, or you can even knock out deleterious genes. For example, cancer-causing genes, if you delete it by this means, you can target cancer cells specifically, and you can kill these cancer cells. That is possible now. So this is done through the 2 nucleus domains, HNase motif and RAFC in the Cas9 protein. Now, what is important here is that you can change the sequence. You can engineer this guide RNA in such a way that you change the sequence of this spatial sequence, and you can target any region of the genome of, for example, in this case, a cell, eukaryotic cell. You can target any region in the genome, and you can bring about change. Either it's a disruption of a gene, or you want to correct a gene, you can do that. So the beauty here is that this technology allows you to pinpoint, you can edit the genome of the organism. So further down, a lot of people have been involved in CRISPR-Cas technology studies now. And what people have done is that, as you saw here, that the Cas9 protein has H motif and RAFC motif. So what people have done is that they muted these motif in such a way that the nucleus activity is inactivated. This inactivation of Cas9, this form of Cas9 is called the dead Cas9 or d-Cas9. What is the importance of dead Cas9 here is that once you inactivate the nucleus domain, it still can find where the target sequence is. So you can utilize this dead Cas9 to find genomic regions depending on the sequence of the spatial RNA. So once you can locate these components, you can utilize these for many, many other purposes. For example, you can tag with effector molecules like transcriptional activators. You can activate genes, or you can inhibit genes, or you can change the genome, like epigenetic modification. You can change the like like methylation pattern on the DNA, or you can look at chromosomal changes, like if there is a change in the chromosome structure, one arm of another chromosome is switched to another arm of another chromosome. You can visualize that changes using this CRISPR-Cas system, and you can do base editing. Now there are RNA targeting CRISPR-Cas system as well. You can use it for chromatin imaging. Lot of applications have come. So one recent around 2019, people have used RNA targeting CRISPR-Cas system for diagnosis purposes, basically to detect whether a particular RNA is there in a sample. This has been, the FDA has approved for the diagnosis of COVID-19 as well. So finally, the CRISPR-Cas system has been used for many purposes. One is the research purpose, to understand the function of genes and how does a gene knockout or mutation behaves in the organism level. People have looked into, and in the medicine, people are looking into drug discovery, and then in agriculture to get better products, crops that people are using it. And for biofuel production, people are using CRISPR-Cas9 nowadays, and even some of the normal entertainment also has been involved. For a few examples, as we talk, as I talk about sickle cell anemia, what people can started demonstrated is that if you take blood stem cells and correct the mutation that I talked about, GTZ to GAG, which cause the sickle cell anemia, can be targeted, and you can correct the stem cell genes in those stem cells, and you can implant again back into the individuals who have sickle cell anemia. You can have a normal life by doing this CRISPR-Cas technology that has been shown in mouse. And then in crops, again, people have utilized this to change the characteristics of many of the crops. Here, for example, tomato, then grapefruit, and other vegetables and fruits have been modified with CRISPR-Cas system to improve some of the qualities. And for drug discovery, people use a lot of these CRISPR-Cas screens in multiple fashion. And now the final, right now, what is being debated maximum is whether you will do the genome editing in human germ-line cells. What do you mean by germ-line cells is that either the egg from the female or sperm from male, or after fusion you get a zygote. If you do the editing here, that is going to affect the whole generation downstream. So this inheritable change should be done or not. That is the big debate now, because we don't fully understand what is the effect of CRISPR-Cas system. However, as you know, in 2018, I think, Dr. He, Jan Kui from China did germline editing, and he targeted CCR5, which is involved in HIV virus entry to the cells. And these babies, called the CRISPR babies, have, I think, is a single mutation in CCR5 in one of the copies of CCR5 in human genome, and he claims that he has that CRISPR baby. But still, there are a lot of questions, and still there are few issues here, especially how to deliver this CRISPR-Cas system to a specific tissue or specific cells is still very debated. It's a challenging technology, technologically. So still that is there. And I would like to stop here and thank you for joining. Yeah. So if there is any question, we can discuss some of the questions. Yeah.
Thank you, sir, for the presentation. Now the platform is open for the Q&A session. If you have any specific doubt, please unmute yourself and ask. Thank you.
Okay. I think there is no question.
Hello.
Hello. Yes.
Yeah, good evening, sir. It was
Yeah.
a
Take
very
this.
great talk.
Yeah.
Yeah, I just, as you said, and when a bacteriophage infects the cell, the
Hmm.
CRISPR picks up part of its genome and then use it as spacer.
Hmm.
So, is that part is like very specific or like any random part?
It's a
The part.
random. It's random. So there are proteins which are involved. Something like Cas1 is very much involved there. So Cas1 and some other Cas proteins is involved in randomly picking. It's not very specific.
Oh, okay. Thank you, sir.
Hello, sir.
Can I?
Yeah.
Siddharth, yeah.
Siddharth? Yeah.
Yes, sir. I have a doubt. Like during when the bacterial genome is embedded in the the bacteriophage genome is embedded in the bacterial genome.
Yes.
Does it undergo replication even if it is in the CRISPR domain?
Yeah. It is replicated just like the bacterial genome.
So the viruses can still replicate.
No, part of the it's now the bacterial genome. Once it is integrated as a spacer, it doesn't have because the genes encoded by the phages are not there. Phages can't replicate inside that bacteria. However, the genome, it's part of the genome of bacteria, so it keeps on replicating and it's transferred to the next daughter cells.
Okay, thank you, sir.
So, what is a typical, Sada?
Yeah.
What is the typical length of the gene that is copied from the virus integrated into the genome?
Yeah, initially, whatever studied in Streptococcus pyogenes is around 24 nucleotide. Generally, it's around
Just
24
the 24
nucleotide.
nucleotide, isn't it?
Yeah, yes.
And does the similar thing happen in humans as well?
In humans, we don't have Cas protein, so nothing of the virus is being integrated into the genome in this way. So we don't have CRISPR-Cas system. CRISPR-Cas system is seen in archaea. Most of the archaea have it, and some bacteria, not all bacteria actually, around 30% of bacteria has this system.
So this gene that is integrated into the bacterial genome of the virus, 24 nucleotides.
Yes.
So they form because the bacteria do not have exons, I mean introns. So do they code for proteins as well?
No. So far, it hasn't been reported because it's the repeats which have seen in case of Streptococcus pyogenes is around 120 nucleotide and another 20, so around 140 nucleotides. So if it is there also, so far people haven't reported yet. That is possible, but so far people have not reported. I think there is no ribosomal binding site as such in the repeat elements. So I think people haven't reported yet any of the protein. And some of the bacteria, it's much smaller. These repeat units, no? It's much smaller.
So that means in principle there is a non-coding region in the bacterial genome in this integrated region, isn't it?
Yeah. It's a non-coding RNA basically. So people were thinking that this can be kind of a transposon or insertion elements. That's what people were hoping for. But surprisingly, this RNA was there. Yeah.
Good. Thanks. Students should be more enthusiastic and ask more questions.
No, not many students.
Sir, I had a question.
Yeah.
Sir, like you said that this if a bacteriophage attacks the bacteria, then it's a part of its DNA get incorporated. So then it's transferred to next generations also. So like this, all the generations, all the upcoming generation of bacteria means are protected against that particular virus.
Yeah, that particular virus. But the beauty of this system is that some of the viruses have proteins which act against Cas protein. So even if that special sequence is there, it can still infect. That is one thing. Another thing is they can mutate without changing the protein sequence. Very often viruses mutate and they can infect the same bacteria again.
Okay, so because there is one method of means from virus gene deliveries are also there. So means if we do gene delivery through a virus, then we can't use the same virus to deliver the genes in the plants or whatever.
Yeah, that is there, but in plants we don't have CRISPR-Cas system, so still it's okay. So generally they don't have CRISPR-Cas system, so there is a different type of immunity. Not this genome editing kind of CRISPR-Cas system is not there in plants.
Okay, sir. Fine. Thank you, sir.
Any more questions, students? Okay, I think no more questions.
I hope there are no more questions.
Yeah.
Here we have come to an end of today's session. It's my pleasure to thank each and every one of you on behalf of Tai and Anvesha. First of all, my sincere thanks to our beloved speaker, Dr. N. Sadananda Singh, for enlightening us with the significance of this remarkable and revolutionary discovery. I'm sure that this talk was extremely helpful to provide you an expert exposition to the research and significance of genome editing. I would like to thank all the wonderful audience present here for your active support and encouragement. Once again, thanks to all. Have a nice day. Thank you.
Okay, thank you. Yeah.